Welcome to Chapter 10. We are now talking about the Calvin cycle. And so let us get started. So the Calvin cycle is similar to the citric acid cycle in cell respiration. The difference is that the Calvin cycle is anabolic. So we are, instead of building up NADP or NADHs and ATPs, we are breaking them down. We are breaking down NADPHs and ATPs that were created during the light reactions to create G3Ps or to create sugars. So we are taking the energy and we are putting them into sugars or G3P molecules. And so it depends on the ATP and the NADPH of the light reactions. That is what fuels it. And so glucose is not directly produced from the Calvin cycle. Remember that we are just building the G3Ps. And then later on, the steps after that, the G3Ps are taken and built into a glucose molecule. And so it takes 3 G3Ps to build 1 glucose. And so the Calvin cycle can be split into 3 sections. The first is right here, carbon fixation. So these steps here. The next is going to be the reduction, which starts right down here. We are reducing NADPH, and then phase 3 is regeneration of the CO2 receptor right over here. So let's look at phase 1. In one lap around, in the stroma, so picture this, we are in the stroma, a carbon dioxide molecule is going to be added to a 5-carbon molecule called ribulose bisphosphate, or RuBP. So here, the RuBP is just like the oxaloacetate from cell respiration. The cycle is going to start and end with it. So the RuBP from the end of the cycle is being attached, or a CO2 molecule is going to be attached to it. And then we have a number of metabolites. We're not going to learn these names. We're not going to worry about too much here, but know that the process starts with RuBP and a carbon dioxide. The enzyme that is creating or taking this RuBP and connecting it to a carbon dioxide molecule is called rubisco. Rubisco is very prevalent in chloroplasts. It's the most prevalent protein in a chloroplast, and also thought to be the most prevalent protein in the world on Earth. So that is phase 1. Phase 2 is reduction. This is where we are creating our G3P molecules. In fact, we're creating 6 G3P molecules. However, so we're taking NADPH. We can see our NADPH. We're taking 6 of these. And ATP are used to create 6 G3Ps, or 6 sugar molecules. However, here's the surprising part, that there's a net gain in this overall set of reactions of just 1 G3P. 5 of those G3P molecules go back into the cycle to refuel it, to restock it. And so 1 net G3P per lap around the cycle. And so remember, it takes 3 laps around the cycle to produce 3 G3P molecules. And it takes 3 G3P molecules to make a glucose molecule. And so this is the single net G3P. So 3 carbon dioxides per 1 G3P and 9 carbon dioxides per 1 glucose molecule. Phase 3 is regeneration of RuBP. So we are going back to, so we've made our G3Ps right here, and we're now going to start making our RuBP again to go back into the beginning of the cycle. So 3 ATPs are used to rearrange the 5 G3P molecules. So those were those sugar molecules that we said are going back into the process. Here they are. So we're using 3 ATP molecules to rearrange those 5 G3P molecules into RuBP, which will then go back into the cycle and be attached to a carbon dioxide. And the cycle starts over again. And so to summarize, the Calvin cycle consumes about 9 ATP and 6 NADPH for every G3P molecule. And the G3P molecules, like we said, go into another set of chemical reactions that synthesize the glucose, the disaccharides, the polysaccharides. And so here we can see the carbon dioxide, the ABT, the ATP, and the NADPH that goes into creating our single G3P molecule. And these are our leftovers. All right, well, that is it for today. I hope that you have a great day.